Okay, we are recording. There's no one here, there's no one online. Therefore, if you are watching this, you are watching the video. Therefore, to get the attendance points, every time I say an attendance word throughout this video, you also need to take a screenshot. So at the end of this, you're going to send me an email that has all the words and all the accompanying screenshots for when I said those words. And the first word that you're going to send in, and you're going to need to take a screenshot of when I say it, even though it doesn't show anything, the word is red, like the color red. That being said, this lecture probably won't start for another 9 to 10 minutes. So since you're watching the video, feel free to jump ahead. And as always, I'll remind you, since you're watching a video, if you look down at the bottom of your screen, there should be a little gear icon, and you can speed it up if you want to make things quicker. We've got about a minute left. So I am going to do attendance. As always, if I don't call your name and you're online, you don't need to worry about it because I'm only right now concerned with who is here in person. So Van Gura is not here. Casebolt is here. Tristan is not here. Covington Griggs is not here. Dow is here, right? Yep. Dow is here. Hughes is not here. Lemke is not here. Carla is not here. Mankins is not here. Mathis is here. Rains is not here. Reed is not here. Elise is not here. Thompson is not here. Yandui is not here. Elkins is not here. So that's it for attendance. We have the 3 people who are physically here. Obviously, if you're online, you're going to need to do the usual things. You're going to write down the attendance words, send those in sometime before 9 a.m. And the last attendance word, if you will, is going to be asking me a question about the material covered today. But as a reminder, some people still, they're watching online, or at least logging in, but then they're not sending the words. So if you don't send the words, you don't get the extra credit points, and you're not counted as present. So you have to send the words. Nothing new, but for some of you, you're still not doing it. Anyway, any questions before we get started? All right, then let's get started. Let me back up. We're not going to do announcements yet. Let's start with where we left off. We left off talking about nucleotides. We talked about the 3 parts of the nucleotide. Come on. Back it up, back it up. Yep. Come on. Sorry about this. Come on. Man, this thing is slow. There we go. This is where we left off. We left off talking about the 3 parts of the nucleotide. And like I said, you're going to need to know this for the exam. You need to know that the 3 parts of the nucleotide are the nitrogenous base, the sugar, and the phosphate. There was a lot of information I gave. Like right here, there's a lot of stuff about the phosphate. Here, there's a lot of stuff about the sugar. Here, there's a lot of stuff about the nitrogenous base. For the most part, you don't need to know that other stuff. There were a few things you needed to know, like, for example, the nitrogenous base sticks out from the backbone, the sugar phosphate backbone. That'll be important later, and that was it. So, again, these are the 3 parts of the nucleotides. This is the last thing we talked about on Wednesday, and this will be the first word for attendance. I'm going to name 2 parts of the nucleotide, and you tell me the 3rd that I did not name. Phosphate group, nitrogenous base, and blank. It's your first word for attendance. Of course, if you're watching the video, that's your 2nd word for attendance. But for the rest of you, that is the first word for attendance. And then, again, the question is, what are the 3 parts of the nucleotide are phosphate group, nitrogenous base, and blank. Oh, another thing I'll mention too, for the exam, I'm not going to try to be tricky with you. So you don't even need to necessarily focus on the phosphate group or nitrogenous base. So when on this multiple choice question exam, I'm not going to have like nitrogenous base or nitrogenous acid or nitrogenous group, right? I'm not going to try to trick you on that. So if you can just memorize, whoops, actually, you know what? I'm glad I paid attention there. For that first one, base is actually the more important part. I'm not going to ask you, so you can remember base, sugar, actually, same here, sorry, guys. Base, sugar, and phosphate would be fine. I guess the point here is, again, you don't need to memorize all those little words. I'm not going to try to trick you. Anyway, let's move forward. So again, these are the 3 parts of the nucleotide. There's 4 different nucleotides, right? Whether we're talking about DNA or RNA, there's 4 different varieties. Just like when we talked about amino acids, right? There was 20 different varieties of amino acids. When we talk about nucleotides, there's 4 different varieties. And they are broken down. Those 4 different varieties are broken down into 2 different categories. There's single ring and there's double ring. Now, the fact that some of them are single ring and some of them are double ring will be important later today in the discussion, but I'm not going to ask you to memorize that for the exam. Unless it's for like extra credit. I don't plan on having extra credit right now. But yes, technically the T and the C are single ring structures, as you can see at the bottom. And A and G are the double ring structures, as you can see up top. And also, even though it's not written on there, if we were talking about RNA instead of DNA, the U, or excuse me, the T, or is that? Yeah, the U is also a single ring. But again, for the exam, you do not need to know which ones are double ring and which ones are single ring. You will need to know, and you don't need to write it down now because I'm just bringing this up. I'm going to teach you officially later. But you will need to know that A always pairs with T and C always pairs with G. You'll learn that later, but that's just a preview. So are there any questions about this slide? All right, some of the questions you're going to get are going to be about comparing, contrasting the difference between DNA and RNA. So they have similar chemical structures, but there are some differences and you need to know them. I've said this before, I'm saying it again. DNA has a T, RNA doesn't have a T. RNA has a U instead of a T. Like I said earlier, even though I haven't officially taught it yet, when we're talking about DNA, A always goes with T, C always goes with G. Now again, since RNA doesn't have a T, for RNA, I'm going to put a D here, for RNA, A would always go with U, and C would always go with G. And that's not trivial, that's going to be important. Again, I haven't officially taught you that. When I say officially, I mean I'm going to have a slide up here where I formally give you that information. But for the exam, you will need to know that. And that's not going to be a question like A always goes with what? Or C always goes with what? It's not going to be like that. What I'm going to do, and I'll talk about this more once we get to those slides, is I'm going to give you a whole list of letters. Like here's a DNA code, you know, all these random letters, and then I'm going to ask you what code goes with it. So I'll give you, you know, like, I don't know, 9 letters, and then you'll have to choose the 9 letters that complement it. But again, we'll talk about that later. Right now, this is just a preview. Any questions about this slide? All right, if you download this PowerPoint, you can watch this little animation that shows a little bit of a difference between DNA structure and RNA structure. Same here, if you download this PowerPoint, you can watch this experiment about the Hershey-Chase, this little short video about the Hershey-Chase experiment, which I won't get into. But that's also a good topic for independent work. So your your online counterparts, you know, not the ones who aren't taking it in person, they have something called weekly discussions. So instead of doing independent work, they have to I give them things to write about and they have to discuss it. Anyway, one of the common themes that they have is one of the questions I ask them is, how do we know what we know? So all these things I'm teaching you, you should think to yourself, well, how do we know that? Who figured that out? When did they figure it out? How did they figure it out? And this is what reminded me, because that's one of the explanations. So for anything I teach you, that's a great independent work topic, right? Like, how do we know these things have 2 rings and these things have 1 ring? Who figured that out? When did they figure that out? What happened? Was it a big, what happened after they figured it out? Was it like a big groundbreaking discovery, or was it just like, no big deal? Anyway, now we're going to get into a little bit more of the important stuff, which is Watson and Crick's discovery of the double helix. So I'll remind you what we talked about on Wednesday. Remember, we started talking about this almost like a timeline. I gave you back in the day, we knew this, then we figured out this, then we figured out this, then we figured out this. And the way we left it off when we were talking about that timeline, when we were talking about the history of what we know, we left off saying that we don't know, in that point in the discussion, we didn't know the 3-dimensional structure of DNA. And that was really important because once we figured out the 3-dimensional structure of DNA, then we know more about its function. So we're kind of going back into that, right? We're going back into this timeline, back into this story, the history of what we know. So here we go. There's this guy's named James Watson and Francis Crick, who were the ones that determined that DNA is a double helix. I'm going to put a question mark next to this. I'm probably not going to ask you the names of who did this. There was other people involved, like Watson. Well, I'll get to that in a second. But anyway, I'm probably not going to ask you this. You can look into it if you want for independent work. They're actually interesting. I need to find this video, the movie. There's a movie made of them and their discovery. It's kind of entertaining. These guys were interesting because they were like really slackers. They weren't like the best students ever. They spent a lot of time at the bars outside of the university, and it just so happens that they happened to discover one of the most important discoveries in biology. But anyway, I'll share that with you. I think it's entertaining. Anyway, they are the ones that determined it's a double helix. What is a double helix? A double helix is a spiral with a uniform diameter made of 2 polynucleotide strands. So again, remember, nucleotides, right? If we're talking about DNA, the polynucleotide is the polymer, and the nucleotide is the monomer. But when we're talking about DNA instead of RNA, it's double-stranded. So when you see, if I were to untwist it, right, it would look like this. but also, and I'm going to give you a better picture a little bit later. It would look like a ladder that has rungs in it, right? Because it's two strands. So when you see that double helix of DNA, this will be important here in a second, that's actually two separate DNA molecules that are together. And we'll talk about what brings them together later. Anyway, like I said, Francis and Crick were the only ones involved. There was someone named someone named Rosalind Franklin who did some x-rays, and she actually inadvertently was a really big help. They kind of stole, I'm going to say stole, depends on how you look at it. Like I said, it's an interesting story. They saw her images, and it's not like they were working together. It wasn't Watson, Crick, and Franklin. Franklin was doing her own thing. Watson and Crick were doing their own thing. And then Watson and Crick happened upon her images, and that really changed what they knew, and they were able to put all the stuff together. A lot of it because of Franklin. Anyway, they used wire models to construct the model, and we're going to talk more about the model. So again, as far as the exam is concerned, there's nothing really important on this slide. This is just kind of like a preview. Again, the old textbook tells it like a story, so I am too. Again, if anything for the exam on this slide, you would just need to know what a double helix is. I'm probably not even going to ask you that, but I might. But again, it is going to be important that you know that that's two molecules held together. And again, like I said, we're going to talk later about how they're held together. Any questions about this slide? All right, here's some pictures. Here's Watson and Crick on the left, looking at their model. Again, that was groundbreaking at the time. There's a picture of Rosalind Franklin. So here we go. The model of DNA is like a ladder that's been twisted into a spiral, just like I described it earlier. So these sides here, the sides of the ladder, right there, the top and the bottom right there, that would represent the sugar phosphate backbone. And the rungs in between, right, that would represent the nitrogenous bases that are connected by hydrogen bonds. And that's going to be important here in a few slides. So I asked you a similar question I think Wednesday. I'm going to ask you a different question, but similar. Hydrogen bonds. Let's see if you can remember this from chapter 2. And I told you in chapter 2, this is going to be important a little bit later, or later in the semester. Hydrogen bonds, are they the strongest of the bonds or the weakest of the bonds? Strongest. Weakest, good. I'm glad you tried. But, so to remind you too, what we talked about on Wednesday, when I told you about the sugar phosphate backbone, I said those are covalently bonded. And I asked you, so there's covalent bonds, the strongest or the weakest. And those are the strongest. So you might be thinking about what I said on Wednesday. So to remind you, the thing holding the sugar phosphate backbone together, that's the strongest bonds. The thing holding each side of the ladder together, so to speak, that's hydrogen bonds, are the weakest of the bonds. That's going to be important here shortly. It's the base pairing rules that dictate the combination of the bases, which is saying, like I said earlier, and I still haven't officially taught you yet, if this side was an A, right? Oops, it'll let me draw it. It won't. If that was an A, then this would have to be a T. If this was a C, then this would have to be a G. If this was a G, this would have to be a C, so on and so forth. There's rules, right? So whatever one side is, that dictates what's on the other side. Any questions so far? All right. Here's just a little picture from the old textbook. Again, just imagine like a ladder, in this case a rope ladder, that's been twisted, right? The twist isn't that important for what we need to know, but the fact that we have two different molecules, each one individually covalently bonded to itself. We have two different molecules, hydrogen bonded to each other. That will be important a little bit later. But the twist, at this level, the twist doesn't really matter. When you get into deeper biology, that twist does come into account, but not here. Here's some more pictures. Here we have a ribbon model all the way on the left. So again, this blue and yellow represents the sugar phosphate backbone, right? So there's one molecule. This one molecule is covalently bonded, right? Those nucleotides are covalently bonded to each other in the sugar phosphate backbone. Same here. This other molecule is covalently bonded in the sugar phosphate backbone. And these two things are held together by those little dots, right? Those little dots represent hydrogen bonds. This is the same thing. We're looking at the same thing, just in a different way here in the middle. That's the atomic model. Again, you can see here, this molecule right here is held together by covalent bonds between the sugars and the phosphate. This molecule right here is held together by these strong covalent bonds, right, between the sugar and the phosphate. These two molecules are held together with each other by these weak hydrogen bonds. And I will point this out, not that you need to know, but again, I did mention that some of them are double ring, some of them are single ring. Just so you know, that is important because a double ring always goes with a single ring. See, double, single, double, single, single, double, single, double. That is important, but not for this course, right? So I'm not going to get so deep with this and the questions that you need to know that. But just so you know, for example, mistakes are made. So if we're trying to build a new DNA molecule, and we're going to talk about that later, and the cellular machinery reads this A, it needs to bring in a T. But if it brings in the wrong one, like for example, if it brought in a G, that wouldn't work for two reasons. First of all, G is double, and it's a double ring, so is A. So then the DNA would get too wide right there, right? It would be too big because you have two double rings. That doesn't work. Another thing that keeps them together properly is you can see here, A has two hydrogen bonds, right? And T has two hydrogen bonds. G and C have three hydrogen bonds. So there's like two different things keeping them from doing the right thing, or keeping them doing the right thing. Because first of all, the double ring always goes with the single ring. And second of all, they all have different numbers of hydrogen bonds, right? And again, you do not need to know that for the exam. You don't need to know that C and G have three and the T and A have two. These are some of the things that keep the machinery doing the right thing. And then one last example I give you. So again, I said A and G would be bad because they're both double rings, so the DNA would be too wide. Remember, form and function, right? Another thing that wouldn't work would be a T and a C, because a T is single ring and a C is single ring. So in that case, in that spot, the DNA molecule would be too skinny. That will be important later, but not for you guys as far as the exam is concerned. And then of course on the right, I'm not even talking about that, but that's the computer model of DNA. So my next word for attendance, or phrase for attendance, if you will, there's three different models of DNA on this screen right now. Which one is in the middle? So that's it. That's the next word for attendance. Any questions about this slide? And just as a reminder, when you're studying, it's not like you need to study these pictures. I'm not going to give you any pictures of DNA and ask you to label the parts. I'm not going to ask you how many hydrogen bonds are between A and T and how many hydrogen bonds are between G and C. I'm not going to ask all that stuff. Anyway, if there's no questions about that, let's move forward. If you download this PowerPoint, you can see the little animation that shows the DNA double helix. Now let's jump back into this timeline like I'm telling a story. Let's talk about the history of this. So they figured this stuff out that I just mentioned. They figured out the three-dimensional shape. And then in 1953, they published a peer-reviewed paper where they proposed the three-dimensional shape, the molecular model of DNA. And then about a decade later, they finally won the Nobel Prize for it. There was Watson, Crick, and Wilkins, who I haven't even mentioned yet. All right, I haven't even mentioned at all. Wilkins, the three of them received the Nobel Prize. Your textbook correctly points out, or the old textbook, I don't know about your new one, but the old textbook correctly points out that Franklin should have had a share of that prize. It's not just the medal, you also get a lot of money. But she was dead. She had died previously, so she was unable to get the prize. If you download this PowerPoint and click that picture of the Nobel Prize, there's a little video you can watch that kind of talks about their Nobel Prize. Anyway, yeah, once again, this is not a history class. I wish they had like a science history class, but this is not one of them. So I'm not going to ask you in what year they published or what year they won the Nobel Prize. This is just for your information. But let's get to the more important stuff here. In this paper that they wrote, that they got the Nobel Prize for about a decade later, Watson and Crick wrote that the structure immediately suggests the possible copying mechanism for the genetic material. So as soon as they figured out that three-dimensional structure, it was like the light went off, like, oh my goodness, we finally get it. Because remember, they didn't know how it happened. At that point, they knew that DNA was the genetic material, but they knew the DNA must be copied so it can get passed down from generation to generation. They knew that the DNA code must be read
to be able to make the proteins that it makes, but they didn't know how that happened until they figured out the structure. Once they figured out the structure, it was like, oh yeah, has to be how this works. And hopefully you guys can get to that. But we're gonna come back to that and see if you guys can guess how the structure implicates or suggests how it works. But first, let's do announcements and reminders. Today is the last day to drop the class. I really think, I hate to say this, but this is like the worst semester I've ever seen. I really think some of you should drop the class. Either that or start showing back up. Like people haven't been here and you've been doing for example, most of you didn't come to lab. And the week prior to that, in these discussions, I keep saying, you have to come to lab, you have to come to lab, you have to come to lab, and people just didn't do it, right? So if you're not paying attention, you might consider dropping the class. If you stay, you should really start turning things around, start coming to class, start watching the videos if you can't come to class. But just so you know, again, some people, like I've already had a couple of people write me and apologize. Don't apologize. I don't take it personally. I don't know what's going on in your life. So I don't judge you. But if you do decide to stay, I'm here to help you. Some of you really need to turn it around, and I'm here to help you turn it around. DNA, the DNA lab that we did this week, it is past due today. Again, like I said, most of you didn't show up. And I said the week prior, I kept reminding you, you have to show up. So if you were not there like you're supposed to be, you're only getting half credit. So you have more to do to make up for the other half. As usual, if you didn't get full credit just because you missed some questions or whatever, you could also do that extra stuff to make up for lost points. Either way, whatever the case is, if you don't know what you need to do, go to the course announcements because it explains it all right there. And if you don't know where the course announcements, well, first of all, when you go there, it says DNA lab, but also if you didn't get full credit, in your feedback for the lab, I left a link to that announcement. So everything you need to make up for those points is right there in the announcement. And of course, if you have questions about that, let me know. I'd be happy to help you. Any questions about the lab that is past due today? Okay, next week's lab. Pre-lab is pretty easy. All you have to do is video and questions as usual, but this time you don't have any pre-lab questions. You only have the video questions. Make sure you do that. Next week's lab, again, is virtual, just like this week, but you have to be here. Unless you hear otherwise from me specifically, I'll never tell you as a class. I will tell you individually, you have to be here for lab. And you might be wondering why. If it's virtual, why do we have to be here? Well, there's a few reasons, not that I need to go into detail, but one of the biggest reasons is people just don't do it. If I don't force them to come to lab, then they'll just like forget about it and not do it. That's one of the big reasons. And the other reason, though, obviously, too, is I'll be here to help you, or the TA. Somebody will be here to help you, most likely me. So any questions about next week's lab? All right. And again, it is virtual, so we do have to be here, but I'm saying, just so you know, if you maybe consider doing it before you have to come in, then it might be beneficial for you. I'll just leave it at that. Anyway, as a reminder, this is kind of a new thing, so this is where I'm reminding you, for you people who are here, I've doubled the attendance points. So if you're here in person, you get 4 points of extra credit a day. If you're online, you're still getting just the 1 point of extra credit. Any questions about that? All right. Again, another reminder, exam 2 is still open for a retake. You can take it as many times as you want. It's not timed, it's open book. Once you're done, you have to email me and say, hey, I'm satisfied with my grade. Can you adjust it? If you don't let me know, your grade won't change because I have no way of knowing that you've retaken the exam. Any questions about the retake? All right, and then once again, on the Friday before and the Monday after Easter, we will be 100% online, both lecture and for office hours. So my next word for attendance, let's see, do something on this slide here. When is the last day to drop a class? Not just this class, I didn't mention that, but I guess, yeah, so I guess to be specific, when is the last day to drop this class? That's the question for attendance. But just so you know, it's the last day to drop any class. All right, any questions? So then let's jump back into this discussion about DNA. Like I said, when Watson and Crick figured out the structure, they were like, oh man, this makes sense. I get it now. Now we can see how it's copied. But before we talk about that, let's back it up a little bit to remind you what we talked about when we talked about mitosis. When a cell reproduces, it has to duplicate the information, right? i.e. the DNA. It has to provide a copy for itself and provide a copy for the new offspring. So if you remember, and this is how we kind of focused on it on the mitosis discussion, right? If you had a chromosome horribly drawn like this with that centromere in the middle, that's what it looks like during G1. Then during S phase of interphase, it duplicates itself, right? So then you have two chromosomes, two sister chromatids, and they're connected right there at that centromere. So this is what we're talking about. What we're focused on is what happens during S phase of interphase. Instead of like back then, you just needed to know what happened during S phase of interphase. Now we're going to talk about how that happens. And again, it was Watson and Crick's model that figured out how it happens. So knowing that, right, if there's, I'll try to draw and see if anybody can guess. That's hydrogen bonds, right? So we have the left molecule covalently bonded to itself. We have the right molecule covalently bonded to itself. We have the two molecules hydrogen bonded to each other. So before I tell you how this happens, can you guess how this might happen? And remember, there's A's, like, right? The A's always have to go, the A's always have to go with the T's. The C's always have to go with the G's, so on and so forth, right? So there's rules that dictate what is hydrogen bonded to what. So are there any guesses on how this process might work? How do you think we can take that one strand, or that one double helix, and turn it into two double helix? How do you think it is that we can read that code that's in the middle? Any guesses?
Computer.
Computer?
Yeah.
That is definitely correct, yeah. We can definitely tell that way. But try to think of yourself like if you were actually looking at a DNA molecule somehow, if you had the ability to do that, what do you think is going to happen to that molecule for us to be able to make a copy of it? That's okay. I don't expect you to know. I just wanted to see if you could guess. So because this strand, right, is covalently bonded to it, it has that covalently bonded sugar phosphate backbone, and so does this strand, right? That thing is strongly bonded to itself. And because these two strands are hydrogen bonded to each other, which is the weakest of the bonds, basically it just opens up, right? When there's cellular machinery that opens up those two strands. That way we can get in and read the code. And that's how it happens, right? Because those covalent bonds are strong. So, I mean, you're not physically getting in there with your hands and pulling at it, but that's kind of a good way to imagine it. Imagine grabbing that double helix and just pulling on it. What's going to happen? Those hydrogen bonds are going to break because those are the weakest, right? So these covalent bonds that keep the individual strands together, those are going to be fine. But when you pull on it, they're going to open up. And that's basically what happens. And again, there's a code. So there's a cellular machinery that I'm going to teach you about here in a second that reads the code, right? So blue is the old strand, gold is the new strand, right? So we open it up. There's a machine that's coming in here and says, all right, there's a C, bring me in a G. I'm going to put a G right here. There's another C, bring me another G. I'm going to put it right here. There's an A, bring me in a T. I'm going to put that right here, so on and so forth. So that's how it works. The strand gets opened up. Those nitrogenous bases are hanging off, and that's the template. Those letters that are hanging off the now opened up strand tell the machine what base to bring in. And that's exactly how it works. There's going to be a test question. I wish I could remember how it's worded. But what you need to know is that when you have two new strands of DNA, right, what is it made up of? Let me reword that. Two new DNA double helices, right? So we start with one DNA double helix, and now we have two at the end, right? And the question is basically asking, what are they made up of? So each one of the new DNA double helix, that's made up of an old strand, represented in blue in this picture, and a new strand. So that will be a test question. So, for example, it's not like you have two new strands on one DNA molecule and two old strands on one DNA molecule. It's not like that, right? For each of the two new chromosomes, we have an old strand of DNA and a new strand. Probably, I don't know, I might use the word daughter and parental. It doesn't matter. So I guess for the sake of study, you should know both. But as far as I remember on the exam, I think I just used the terms old and new. Either way, again, when we start with one chromosome, then during S phase of interphase, we now have two chromosomes, technically sister chromatids, But for this discussion, who cares? And for each of those new 2 DNA double helixes, again, one strand is old, one strand is new. Any questions about that slide? Okay, if you download the PowerPoint, you can watch this little animation that shows it in action. This is really cool. I really like it. I will warn you though, if you watch these, there's a bunch of them that I've embedded in this PowerPoint. If you watch them, they are not geared for you guys. So there's going to be a lot of terms that are like, what is that? There'll be a lot of words you haven't heard of, and don't worry about that, right? So if you see any words or terms that I didn't say or that you didn't read in your textbook, don't worry about it. This is for higher level courses. But anyway, it is interesting to watch. It really shows it in action. This one's a lot more simplified. Another thing you can download and watch that shows it in action. The last one was more realistic because it shows the actual 3-dimensional shape of all the molecules. This one's better for like understanding at the 100 level, like how things work. Same here. There's another animation. Again, words that you don't even need to know. For example, lagging strand and template strand. I didn't teach you that. I'm not going to teach you that. Primase. That's an enzyme that does something that you don't need to worry about. There's helicase, which is not shown. The name's not shown there. Anyway, there's all these enzymes that you don't need to know about. So again, if you're watching these videos and you see words that you don't know, don't worry about it. All right. So let's talk about enzymes again. So to remind you, enzymes always end with ACE, right? So if you see ACE at the end of it, you're like, oh, that's an enzyme. And the enzymes, remember, are named after either their substrate or the job that they do, right? So for example, ATP synthase is the thing that synthesizes ATP, right? You learned about that in photosynthesis and respiration. What is it?
Lactose?
Yeah, lactase, right? Lactase is the enzyme that breaks down lactose into glucose and galactose. So, before I show you on the next slide, see if anyone can guess. What do you think the name of the molecule is that polymerizes DNA? So it makes the DNA polymer. What do you think the name of the enzyme is that polymerizes DNA?
Polymerase or something.
Very good, yeah. DNA polymerase. And you need to know that for the exam. Heads up, there's going to be another polymerase you need to know, and you'll need to know the difference between the two of them. But yes, DNA polymerase. That is the thing, when I kept saying the cellular machinery, it's opening up the DNA, and it's reading the code, and it's bringing in the new nucleotides. That's the thing that's doing it, DNA polymerase. Now, I'm not going to test you on this, but just so you know, this is actually an oversimplified thing. DNA polymerase is not actually just one protein. It's actually it's a team of proteins doing a lot of work. And if you were taking a different course, you would know all about those different proteins. But here, we're just considering them just kind of one big cellular machine, and we call it DNA polymerase. And I only mention that because for whatever reason, if you have to do some research on your own about DNA duplication or DNA replication, and you're reading something like, he never said anything about this. He was lying. No, I'm not lying. I'm just oversimplifying. Anyway, this DNA polymerase, this is the thing that makes the bonds between the nucleotides of the new DNA strand. So, and I'm going to come back to this slide, so don't worry. I just want to show you this very quickly. This first bullet point right here, what I mean here is this. This is the thing that says, all right, there's an A right here, so I'm going to bring in a T. I'm going to covalently bond that T to this G. Just like previously, it covalently bonded this G to this G. And this G to this T, and this T to this C. That's the thing that's bringing in the new nucleotides and covalently bonding them to each other. Yeah, like I already said, it does it. It follows the rules of A to the T and C to the G, right? That's all it does, right? Or excuse me, that's what it does. Just so you know, and I'm not going to ask you this on the exam, but it is fast and accurate. How fast? It does 50 nucleotides per second. It's pretty quick, and it only makes 1 in a billion. For every billion bases that it brings in, it only makes 1 mistake. Let me rephrase that. Not that it matters, so I'm not going to test you on this, but I think a better way of saying that is, out of every 1 billion bases that it deals with, you only end up with 1 mistake. And I say it that way because technically, as you can see in this next bullet point, it also fixes mistakes. So it might make more than 1 in a billion mistake, but it usually it fixes them. most of the time. So 1 in a billion times it doesn't fix them. But yeah, so DNA polymerase, not only does it build the new DNA, but it also helps repair damaged DNA, right? So if you get sunburn, for example, or anything else, right? If you damage your DNA, that's what DNA polymerase does. Obviously, it's not perfect. Otherwise, people wouldn't be getting skin cancer from UV light. Otherwise, people wouldn't have died from Chernobyl, right? If it worked perfectly, but it doesn't. It works, but it doesn't work perfectly. So anyway, any questions about DNA polymerase? Again, here's the picture I'm showing you. So even though you don't see it, you have to imagine there's this big molecule that's in this area that's opened it up and it's reading the strand and it's bringing in the new nucleotides and it's covalently bonding those new nucleotides to each other. So let's talk a little bit more about DNA replication, as you already know and as you're going to need to know again for this exam. So you needed to know it for the last exam. Or is this the same exam? Anyway, S phase of interphase, right? That's when DNA replication happens. You already know it, you still need to know it. Something new that you don't know is where it happens, right? So if you have this big old strand, this big old huge chromosome, right? You've got 23 pairs of them. You have this huge chromosome, and the place where it starts, this process starts, is called origins of replications. I might ask you that. More than likely, I would probably maybe just use that term in a question that's not necessarily about that term. But either way, you need to know what it is. So imagine again, it's a horrible picture. Imagine you have this huge chromosome, right? Really long. It's not like the DNA machine, the DNA polymerase starts here and works its way down, right? Or it's not like it starts here and works its way up. Not really down or up. What happens is, along the whole strand, there's multiple origins of replication. So that machinery starts there, and then it kind of works its way out from left to right. Well, left to right in this picture. And then we have these things called, well, it doesn't matter. We don't need to get into it. Anyway, the place where it starts is DNA origins of replication. So the point is, that's what it's called. And just so you know, again, it's not like it starts on one end and works its way down. There's just multiple spots, and then it just kind of works its way out. You get these bubbles until finally you have two daughter DNA molecules. Which makes sense, right? So imagine if I asked you guys as a class, and I said, all right, as a class, and you guys can decide how many people work on it, I need you to write down the textbook by hand. Right? So it wouldn't make much sense if you got math, let's just get Mathis to start at the beginning, go to the end. That wouldn't make sense, right? Like you start on chapter 1, you start on chapter 2, you start on chapter 3, you start on chapter 4, right? You guys would all work on it simultaneously, it'd be much quicker. Same thing here. For independent work, you can look this up though. This, I'm talking about eukaryotic. I'm talking about like our kind of chromosomes. So ask, for independent work, you can look up, is this the same way it works for bacteria? Because they don't have one long DNA, right? They have a circular DNA. So you have to wonder how it works for them. Any questions about this slide? All right, if you download the PowerPoint, in this case, I really do recommend watching this. Because when we talk about the origins of replication and how it kind of works out from that point and spreads, like goes this way, it's really hard to picture unless you see it animated. So I really recommend going to WVSU Online, opening up this PowerPoint, finding this slide, and watching it in action. Actually, I don't remember how long it is. Maybe I'll just show it if it'll let me. And it will not. All right. Anyway, same thing with this. The next slide also shows it in action. Same thing with this. It also shows it in action. Again, if you see terms that you don't know about, like 5' or 3', or lagging strand or leading strand, you don't need to worry about that. But yeah, you can really see it in action. I love it. You can see here that it's opened up. The original DNA has been opened up and split, right? So for each of these old strands, we're adding a new strand. So then eventually when it's done, we're going to have 2 new DNA molecules. Yeah, so we're done with the first major bullet point. That's all about DNA replication. Unfortunately, that was kind of the easy part. Now we're going to get into the slightly harder stuff. And in my opinion, more important part. So now you're going to learn how we go from DNA to RNA to protein. I say this is important because there's a lot of stuff right now where it's controversial with people, and there's a lot of misinformation out there, for example, about mRNA vaccines. You'll know more about how that works once you know this. GMOs, right? You're going to know more about that after you know about this. So let's jump into it. DNA to RNA to protein. And let me say this, and I've said this before, and I'm bringing it back. For this particular topic, depending on how your brain works, the best way to study this might be big picture and then start adding details. So the first thing you need to memorize in this example is that we go from DNA to RNA to protein. Memorize that first of all. That will also be a test question. It's multiple choice. Like, which of these in the correct order? And some of them will be like protein, RNA, DNA. That's not right. RNA to protein to DNA. That's not right. RNA to DNA to protein. That's not right. DNA to RNA to protein. You need to know that. That's the most important thing. If you don't know it goes in that order, then who cares about all these other things I'm about to teach you? There's a name for when we go to DNA to RNA. You'll need to know it, but if you don't memorize the fact that first you go from DNA to RNA, then to protein, then who cares what the name of this process is, and what the name of this process is. And there's all these different parts that are involved from going here to here and going here to here. There's the names of all these different little steps. You're going to need to know all that for the exam, but again, if you don't memorize this big picture, who cares about all the details? So, that being said, let's jump into it. As you already know, this next slide is just a reminder of things you've been taught before. DNA provides instructions to a cell, right? So again, DNA is instructions on how to build protein. Proteins are involved in everything, right? Because remember, every single chemical reaction that's happening in your body is catalyzed by an enzyme. So without that enzyme, that chemical reaction is not happening. And the enzymes are proteins. So that's how DNA provides instructions to a cell. Also, if it's providing instructions to a cell, it also provides instructions to an organism as a whole. As you already know from previous previous chapters, the genotype that we learned about last week is the heritable information. It is the sequence of nucleotide bases in the DNA. That part's new. We haven't officially said that yet. Well, we kind of have. We've hinted at it. The point is, whether you have a chin dimple or a smooth chin, like we talked about last week, that is based on the sequence of the nucleotides, like whether it was AACTG or TTCGA, whatever it may be. It's those letters that dictate whether you have a smooth chin or a dimple. And that's what we're going to learn about in this section. And of course, again, it's the phenotype that results from the genotype. So the question then is, how does this happen? How is it that these letters dictate whether you have a smooth chin or a dimple chin? How do these letters dictate whether you have freckles or not, or blue eyes or not, or hitchhiker's thumb or not? All these things, right? That's what we're going to learn about. All right, so really quickly, I should have done this on the last slide, but it's still here. I won't do that yet. All right, so the first portion we're going to talk about is how an organism's genotype determines its phenotype. So this is pretty good. This is what you already know, right? You already know that the genotype determines the phenotype. You already know about complete dominance and incomplete dominance and codominance. But now we're going to kind of look at it from the molecular level, figure out how it happens. So to remind you, a phenotype, that's the physical traits, right? Whether it be something you can see or otherwise, right? It could be a chin dimple, it could be a disease. The phenotype are the physical traits that are from the actions of a wide variety of proteins. So here, you already know what a phenotype is. Here, we're just describing it in a different way because we're looking at it from a different context. Now we're looking at it from the molecular level. And what we're saying here is that it's proteins that make your chin dimpled or not. It's proteins that make your tongue be able to roll or not. It's proteins that determine if you have achondroplasia or not. Like all those things you learned about, those proteins that were involved. Because again, proteins are enzymes are always proteins, right? And it's these enzymes that control everything in your body. Anyway, any questions about this slide? I put a little star here. Let me see what I try to remind myself something. So a non-textbook example of an enzyme. Here we go. All right. So for example, you can look at even the few people we have in class today. You can look at our skin color and it's different, right? Actually, we're not that much different, but we are slightly different. We have slightly different skin color, but that's not necessarily due to a protein, right? And here we are talking about protein. But there's a gene called the TYR gene, and it provides the instructions for making an enzyme called tyrosinase. Am I saying that right? Yeah, tyrosinase. And this enzyme is located in these things called melanocytes, which are specialized cells that produce a pigment called melanin. So melanin, that's the thing that gives us our color. And melanin is not a protein. So again, even though it's the proteins that dictate all these things, melanin is not a protein, but there's an enzyme that makes the melanin. So that's how it's a protein that dictates our skin color. Anyway, let's keep moving if there's no questions about this. Like I said, what you need to know, the big picture, is DNA to RNA to protein. You have to know that. Now let me add the next bit of information that you should memorize. Once you've memorized that we go from DNA to RNA to protein. Going from DNA to RNA is called transcription. Going from RNA to protein is called translation. You need to know that. You need to know the difference between the two. It sucks that they sound similar because that could be confusing. But once we really get into it, I'm going to teach you ways where you can differentiate between the two and ways to memorize. Transcription is from DNA to RNA, and translation is from RNA to protein. The relationship between genes to proteins is a relationship of information flow. That's just an introduction. I'm not going to ask you that. And again, as you need to know, I've said this a million times in lab, in lecture, the function of a DNA gene is to dictate the production of a polypeptide. And as far as we're concerned in this conversation, a polypeptide is a protein. And I'll remind you what we talked about in chapter 2. Technically, a polypeptide is just a bunch of amino acids put together, right? Technically, it's not a protein until it's folded into its correct shape. But for this discussion and this chapter, whenever you hear polypeptide, you can go ahead and think of protein. But again, technically, it's not technically a protein until it's folded up into its correct shape. But as far as we're concerned, in this conversation, it's the same thing. So again, for the exam, you need to know, big picture here, we go from DNA to RNA to protein. Now I'm adding a little bit more information. That process of DNA to RNA is transcription. That process of RNA to protein, or polypeptide, whatever, is translation. So for the next word for attendance, when we go not from DNA to RNA, but from RNA to protein, what is that process called? There's one word, there's a one word name for that process, going from RNA to protein, or polypeptide. What is that process called? And then, let's see, let me take a look, see if I'm going to talk about this, because we only have like 2 minutes and some change left. Yeah, that'll be a good place to stop. We'll just stop a little bit early. So remember, the last phrase for attendance, if you will, is to send me a question based on this material. You can send me a question about whatever you want, but for the points, the question needs to be based on the material, right? So, for example, I said something about independent work a lot today. Don't ask me a question about independent work. You can, but for the points, the question about independent work won't suffice. You need to ask me a question about the material presented. And that's it. You guys have a good weekend. I'll be here on Monday. Actually, and I'll be online until noon for end of office for office hours.